Welcome to Going Antiviral, the podcast for the IAS-USA, a professional continuing medical education organization focused on HIV and other viral diseases. I'm Dr. Michael Saag, professor of medicine and infectious diseases at the University of Alabama at Birmingham and volunteer member of the IAS-USA Board of Directors. Welcome to the Going Antiviral podcast. Today, March 3rd, 2024, we're at the CROI meeting, the Conference on Retroviruses and Opportunistic Infection, and they're speaking to Dr. Fabian Otte. Dr. Otte is a molecular biologist working on the development of the next generation SARS-CoV-2 vaccines. Is at the University of Basel in Switzerland. Welcome.
Good morning.
Good morning. So, you have been working on a different type of vaccine, not just a standard mRNA vaccine, but one that involves using something called single-cell development process. Can you explain a little bit of what you're doing with the single-cell approach?
Yes, so in principle, it works like this. So we take the viral RNA, we convert it to DNA. And since the SARS-CoV-2 genome is very large, 30 kb, we subdivide it into 4 fragments. And these 4 fragments we then transfect into cells, we bring them into cells. And inside the cells, since they have some overlaps, they will find these overlaps and will recombine. And then you have a full-length virus, basically. So that's how the wild type would work. And for the single cycle, we have one essential gene missing. So one of the fragments is missing an essential gene. And this essential gene is in the cell that we try to use for production is transcomplemented. So it is in the cell, it's producing this gene or protein all the time. And when you come in with these 4 fragments, one fragment missing this essential gene, on the protein level, all proteins are there. So you have full assembly of a viral particle. But in the end, this genome that carries a defective gene will be incorporated. And then if you take this virus for vaccination, it will enter the cell as a normal virus, will then do a wild type replication in the cell. But since this essential gene is missing, the virus cannot assemble, so it's stuck in the cell. But still you have high production of viral proteins and a very broad production. So compared to mRNA, there's only one, so it's always spike. And we have basically the whole virus. Therefore, we expect to have a much broader and longer lasting immunity.
That's great. So translated in a way, it's a live virus attenuated in some way. Not really. It's not really replication competent. Is that accurate?
Well, it's difficult to phrase. It can replicate inside the cell, but it's defective in spreading. So it's not It's not a propagating virus. It's just a replicon virus.
Got it. And so unlike, let's say, the polio inactivated virus, you don't see any way that it could pick up that critical gene just out of nature. It just won't happen.
So we've done these experiments 1,000 times, and we also passage the virus. We never see that is like kind of going back to wild type.
Yeah.
So you could imagine if you vaccinate and you have an infection, wild type infection, then there could be, of course, a potential that these 2 viruses meet. But in the end, then you have a recombination. You still have wild type infection, and this is what you would have anyway. So, yeah.
Got it. So this notion of the 4 fragments going together, is that what you're referring to as the clever approach?
Yes. So clever is basically you going from RNA directly. So you do a reverse transcription step. So you have then DNA, and this DNA you then transfer into the cell. And therefore you would not have to clone anything. So you just put into the cell, and then you have, for wild type virus, you have, I guess for some of the virus, we have even after 2 days post-transfection, we already have infectious virus. It's very fast. But it comes hand in hand with, since you're doing an RT step, there will be, of course, sometimes some mistakes happening. And it's not 100% perfect, but still it's very fast. And to overcome this, you can also go and clone the virus and then use very high fidelity polymerases to amplify the region of interest. And since these high fidelity are super, super good, they will not have any mistake. And in the end, also only the fittest virus will come up, and it's most of the time the virus you expect to come up. So it's very, very easy to do.
Right. So you move forward then, and you've used a hamster model to do a intranasal challenge with this product. What have you seen in those experiments?
So first, it's very important. So we do not inject into the muscle. We apply it through the nose. So we do prime boost
with a swab.
It is so. I have not done the hamster myself. So there's a collaborator in Germany. But what they do is, I guess they how you call it, hibernate. So that they fall asleep basically. Yeah. Then they apply per nostril a certain amount of virus, so that it is in the natural route basically, and then they wake up, these hamsters, and then they come into contact. So 24 hours after or that infection, but vaccination, they come into close contact with these contact animals, and then you can study: is there any potential threat, as you mentioned, that there is a conversion back to the essential gene is missing, and then you can study the spread of the vaccine virus. And then afterwards, when you do the challenge, you can study: is there any challenge virus transferred to the contacts? And when we look into the data, the most advanced candidate, we do not only take out an essential gene, we also take out genes which are important for the virus to hide, basically. So they, for example, one very interesting aspect is it has so many genes invented basically to overcome interferon signaling. It's super afraid of interferon. So we take out genes involved in interferon signaling, as well as MHC1 downmodulation. So MHC1 is important for killer T cells to basically act. And these genes we take out. So we hope that we have then a much more immunogenic vaccine, which could even outcompete a wild-type infection, because many people come to us and say, well, having a wild-type infection or having your vaccine, what is the benefit in the end? And the benefit would be then that you have these functions taken out, and therefore much better signaling, and hopefully therefore also a longer lasting immunity.
So in essence, these genes that you've taken out are precisely the ones that make COVID-19 so dangerous in a way, right? Because the genes that are protecting the virus, that it hides from the immune system, that it fights off interferon, that it can be longer lasting in some people, especially if they have an impaired immunity. Those are the things that you've actually stripped away on the candidate vaccine, and interestingly makes it more immunogenic and therefore a better vaccine candidate.
So I imagine that it is in the first days, it's very immunogenic. On the other hand, when you look later, so that's what you also see in the hamster, that this inflammatory response is reduced. So also in the light of post-COVID or long COVID, could be a very interesting thing to study, because if we would have first a very good immune induction, and then which gets then really rapidly shut down, hopefully those people that could suffer from this long COVID have then a potential that they will not have in the end, long COVID or post-COVID syndrome.
And in the hamsters, once you've done the experiment, you've vaccinated, you've exposed them, then you watch for immune response and you measure that in different ways, T cells, other things. What have you seen in your immunology studies?
So what was very striking is when we challenged the vaccinated animals, they were protected. This is for mRNA, it's the same, but the difference between mRNA and our vaccine is that there was no spread to the contact animals. So they are in very close contact. They sniff at each other, so they like to having partners, and then this way they transmit it very easily in the hamster model. Also, one aspect, maybe just to mention on the side, you just need 5 infectious units for the hamster to have a full-blown immune response, and therefore it's a very sensitive system. And when we did that, there was no transmission. So this is what you want to achieve, right? Because for mRNA, you are protected, but the people around you that might not get the vaccine because they suffer from immune compromised situation, they cannot get the vaccine, and these are the vulnerable populations that need protection most, and our vaccine would work this way. And even our vaccine could be used to vaccinate also these vulnerable populations, because we have a very safe vaccine. And coming back to your question regarding the immune induction, we also measured in the lung tissue secretion of certain cytokines. So one cytokine which is important for viral or microbial infections is interferon gamma. And this one we monitored. What we could see is that for wild type, the inflammatory response was high for interferon gamma. And for our clinical candidates, they were very much reduced. And also another cytokine which is important, this is like the counterpart of interferon gamma, because too much would be bad. So IL-10 is one that down-modulates then this inflammatory response. And this one was also rather reduced. So we have seen that the immune induction is reduced. And on top of that, we also used peripheral monocytes, blood monocytes. We isolated them from patients that had already had either disease or were vaccinated. So they have already seen something from the virus. So we isolate these PBMCs and we fish out the interesting populations. And in the end, we stimulate these cells with viral particles, either our vaccine, single cycle vaccine virus, or the wild type, and also negative control, of course. And then what we saw is that especially in the memory compartment, which is the one that will respond, we saw that there's a much better induction. So already after 1 day of exposure of interferon gamma, that's what we think is the effect of taking out 6 and 8. And we also see, especially in memory cells that are activated, we see that the T cell receptor, if this gets down modulated, this can show you that there's internalization and there's potentially a cognate antigen present. This is the case why it's down modulated. So you see in the negative control as well as the positive control, CD3 is much higher compared to our vaccine virus. This is hopefully showing us or indicating that there is an active signaling or much better signaling. And this would be the T cell part, which most of the time is neglected because all the people most of the time just focus on the antibody response, which is important. But the other side of the story is also important, the T cell.
So this downregulation in the T cell memory cells of the receptor is in essence a way that the immune system sort of gets a stand down response. When it has an infection, it's done its job, and then it says, okay, you go back home, you can rest now. That sort of gets screwed up in a lot of normal SARS-CoV-2 infections, right? And that's what causes the severe symptoms from day 7 onward in a lot of people,
yes.
Right? So this is pretty exciting. So the question I'm sure everybody's wondering right now is, what's next? When do you go into humans? Or first off, let's go back. How easy or difficult will this be to scale up?
Mm-hmm. So in our case, the vaccine has the benefit that in the production system, we do not in the end attenuate it. So others, they just take out certain parts of the virus, or some other aspects, they call it codon de-optimization, that the virus uses codons that it doesn't like to use. And this is how they slow down the virus. But slowing down the virus also means production-wise, in production system, the virus is slower. And for manufacturability, this is a bad thing, because best thing would be to have the highest titer possible, because it's the lower cost then. That's one aspect. So when we do our system, we are at the wild type range levels to achieve these high titers. And we can basically, for these single cycle viruses, there are certain components that you have to have in a cell, but with the virus you still can fiddle around. So let's say the vaccine efficacy goes down because there's a new variant, of course, coming. You can play with this clever system. You can play like Lego stones. You can just take out S, replace it with another S, or if there's another important function that you want to take out, you can easily manipulate it and then put together the virus and have then in. So we have recently made an Omicron XBB.1.5 adapted version, which we just had from scratch. It took us maybe 1 month to have it. And this would be, of course, very powerful. Of course, if you compare to mRNA, mRNA is super fast. But on the other side, the downside is you have only one single protein that is doing then the job. And for our case, it's much broader. We're maybe a little bit not that fast, but still very fast compared to others. And I think this can be very important.
And when you say the S region, I'm assuming that's the spike protein.
Spike, yes.
And so that's what you're shuttling because that is responsible for the variants as they emerge. So, and you're also giving, as opposed to mRNA, you're giving it intranasally. And so I imagine that would get more mucosal immunity.
Yes.
And have you guys measured in the hamsters IgA? Do they even have IgA? I guess they do.
So the thing is, the hamster model is not that developed yet. So at the moment, we are running experiments in the mouse. The mouse model on the other side is also not the best model because if you have, for example, live attenuated vaccine, and if you overdo it, they have brain pathology. So all of the mice will die. So first round that we want to test is if we apply a high amount of our virus for the nose into these mice, do they survive? And do they have any pathology in the brain? Because that's a big issue in the live attenuated vaccines, if you can imagine. And we will work on that. And
So you've got to come up with the proper infectious dose or exposure dose, I'll call it, for the vaccine to be administered to humans as you move forward. So it's production, scale up production, getting it right, getting good manufacturing processes where the product is uniform, and then start testing dosing to make sure you don't overdose.
Yes, so we would have also, because you have to look for regulations, you have to do the tox studies, of course, scale up, and you also have to, the dosing is also important, of course. What we see is the highest dose we can achieve, and we put into hamsters, they have no loss in weight. So when the hamsters get sick, they lose weight because they don't take up food, they feel really sick. This is something you can easily monitor. And for us, we did not see any loss in weight. This was really interesting. So they tolerate the vaccine really well. And one other aspect of coming back to the manufacturability is that we, compared to mRNA, we can, for example, lyophilize the virus. That means freeze dry, and we can store it as a powder. And this is easy to distribute then. And also you can just then, reach, let's say, rural settings, and they just have to add a certain amount of distilled water, and then they can have the vaccine shots. So this is also one aspect that mRNA is very bad at, because they do not have, they have to be cooled down to -80. And that's
I think the public is familiar, who were paying attention back in December of 2020, about the cold chain.
Yes.
And with lyophilization, you don't need that.
Yes, so we have to,
or at least not as much.
We have to do much more experiments. But what we see is, if we store it at 4 degrees for a long period, there's no loss of infectivity of our single cycle virus, which is very, very helpful. And we hope to then have, of course, then again, another advantage compared to mRNA.
So the time has gone by quickly because it's so interesting. But before we close, I want to just get your view of what could be next with this approach for other viruses, other RNA viruses in particular. Dengue, you mentioned chikungunya.
Yes.
What are your thoughts there?
So, of course, this is kind of an idea, platform idea. We also think about, can we commercialize that, or can we work on towards like a startup, something like this? So the technique for chikungunya and for dengue we tried, and it was just one shot, and it worked immediately for the wild type. For the single cycle, it's a little bit more difficult. You have to spot the gene that is giving the virus difficulty to replicate, and also in the end, this gene has to be transcomplemented in the cell, and it should not be toxic to the cell. If it's toxic to the cell will die, and then you cannot use it. So this is the difficulty. And but you can, in principle, it should work also for other RNA viruses. Yeah.
Something I meant to say, as I thought about with dengue, is that with that, there's a special challenge that sometimes a second exposure to dengue can lead to the hemorrhagic fever. And that obviously is the fatal version. And so there's concern that if you give a vaccine and someone gets exposed, you could trigger that. That's a whole different set of complications than you might see with SARS-CoV-2.
Yes, so we talk to a lot of experts, and whenever we present our data and we say, well, we could use it for chikungunya or dengue, most of them say, well, you should start with another virus, not dengue, because dengue is super complex with these antibody enhanced.
There's 4 serovirus, you've got to get all,
Yes, that's why they say you should rather maybe move forward another candidate. And dengue, you can still think about it, but it's difficult.
Well, this has been fantastic. And what I've learned along the way is that there's this single cell approach that you're using can not only create a candidate SARS-CoV-2 virus, but that in hamsters it's protective even when the transmission is so efficient in hamsters, and that the downregulation of the immune response in the sense that once stimulated, there's the appropriate production of innate immunity, but then it gets downregulated. I did have this one final question, and that is, if that's going on, do you have any sense of the durability of the vaccine if you were to use it? And I know we can't say humans, but in the hamsters, for example.
So in the hamsters, we just did very short periods in between the prime boost and the challenge. Of course, the next step would be to look also how long longevity of protection. This is what we should do in the near future. But at the moment, we do not have the data for that.
Right. And also, we're going to be giving this, if it makes it to market and people start using it, I would bet by that time, most everyone has had a SARS-CoV-2 infection, at least one. And they've had prior immunity, a lot of them with mRNA vaccines. So studying in those populations is going to be important to make sure that it's feasible and that it works.
So our vaccine would basically be then always a booster. So we did it with naive animals, and then the next thing to study is, of course, how does this behave in the system where you have already pre-exposure to SARS-CoV-2? Do we have a benefit if we use our vaccine compared to the wild type? And these are studies that have to be done at some point.
Fabian, it's been a joy talking to you.
Thank you.
I've really enjoyed it. I think what you're onto has got a lot of exciting promise, and I wish you all the best for successful continued development of this exciting vaccine.
Thank you very much, and thanks for having me.
Thank you for listening to this episode of Going Antiviral. Catch up on earlier episodes wherever you listen to podcasts, and check out the video versions of this episode and others on the IAS-USA YouTube channel. You can find these links in the show notes, or simply go to YouTube and search for IAS-USA, and there they are. Be sure to subscribe, rate, and review Going Antiviral on Apple Podcasts. This podcast is for informational and educational purposes only and is not intended to serve as medical advice. Opinions expressed in this podcast do not necessarily reflect the opinions of the IAS-USA or its affiliates. If you have a question you'd like answered on the show, you can send it to podcast@iasusa.org to be answered in a future episode.